Well, hello, gang. This is Mr. Mormon again. Welcome to Lesson 3 in the Nature of Science. We're continuing talking about the scientific method, and today we'll focus on experimental design. When talking about experiments, there's certain terms or vocabulary words that are often going to come up, words and terms that you definitely need to know and understand. Now, the first three, observation, inference, and hypothesis, we have already defined. These are in previous notes. You can just go back and look up those definitions if you need them. These are some additional vocabulary words that we need to define and get into our notes. A theory is a broad explanation of natural events that is supported by strong evidence. In science, theory means something different than we use the word in everyday language. In science, a theory is an idea that's been proven over and over and over again. It's very well supported and it's very strongly believed. In everyday language, we use the word theory to mean, that's my idea. It's what I think. But in science, it's a very strongly held belief with lots and lots of data, lots and lots of experiments to back it up. Evolution is a good example of that. Evolution is referred to as a theory, and some people kind of discredit evolution because it has that word attached to it, thinking that it's just somebody's idea, Charles Darwin's idea. But it's much more than that. A theory has been backed up, and there's lots and lots and lots of evidence to support it, and it is strongly believed by the scientific community. Next is controlled experiment. A controlled experiment compares the results of an experiment between two or more groups. The experimental group is the group being tested or getting the treatment, for example, a new drug. The control group is the normal group. This group is going to be treated exactly the same as the experimental group. It will be identical to the experimental group, except for one difference. It does not get the treatment. This group would receive no drug at all, or maybe they would be given the original drug, not the new drug, or a placebo, like a sugar pill that has no medicine in it. But they're told that they're being given the experimental drug. That's a placebo. And then finally, there are constants. They're also sometimes called controlled variables. I try to use the word constants all the time in class so as not to get them confused with the control group or a controlled experiment. Constants are something very different. Constants are all of the factors that are kept exactly the same in each setup of the experiment to make it a fair test. The only thing that should be different in your setups is the independent variable, the factor you are testing. We could also call that the manipulated variable, and that was all covered in a previous video. Okay, now let's write down some of the characteristics that make a good experiment. A good experiment is repeatable. It could be repeated by anyone and they would get the same results. A good experiment will have a large sample size or many test subjects. This is something that comes up on both the state test and the living environment regions. They will often ask, how could you make an experiment better? And it's always a good answer if you could figure out a way that you could collect more data, have more test subjects, or test each subject more times, more trials. Increasing the amount of data is always going to make an experiment better. Along these lines, a good experiment may be performed over a long period of time. They will test only one independent variable. This is super important to remember. A good experiment will only have one variable. If you test more than one thing in the same experiment, you don't know which variable caused the results. A good experiment will be peer-reviewed, meaning it's examined and repeated by several scientists to determine accuracy. This goes back to what we were talking about with communication. Scientists publish their work. They put their work in scientific journals so other scientists can not only see what new research is being done, but they can try it themselves and confirm that the results are valid. A good experiment does not have to agree with the hypothesis. Um, a scientist's prediction is allowed to be incorrect, and it often is. Doing good experimentation, collecting good data, making good observations, that's the important part. Um, your hypothesis is what you thought the answer was before you did the experiment. The experimentation and the data collected and how carefully you collect it and record it and report it, that's what makes a scientist a scientist. It's not whether how many of their hypotheses are correct. Proving, like we've said before in earlier videos, proving a hypothesis wrong is often just as valuable as getting support for that hypothesis. It helps lead you towards the truth, which is the goal of science, to get answers, to solve problems. And finally, a good experiment is going to be objective. The experiment and conclusion will be fair and unbiased. Fact and opinion are not mixed. Okay, that's it for the notes for today, but I do have a couple of examples of questions, one from an 8th grade state test and one from the living environment regions that I'd like us to look at. Okay, this question comes from the June 2013 New York State Science Assessment for 8th graders. I don't know if you're able to read that or not, so I'll read it to you. Base your answers to question 46 and 47 on the diagram below and on your knowledge of science. The diagram shows a controlled experiment designed to test how much time it takes for seeds to germinate under 4 different conditions. 4 bean seeds were placed in each of 4 pots. Each pot contained 100 cubic centimeters of soil. All 4 pots were placed on the same sunny windowsill. A different amount of water was placed in each pot. And you can see they have it labeled right below each pot. 0 milliliters of water, 10 milliliters, 20 milliliters, and 40 milliliters of water. Question 46 asks to identify the independent or manipulated variable in this experiment. What would you put as your answer? Okay, if you said it's the amount of water that each gets, you're right. That's the one thing that we are changing or manipulating in this experiment, the amount of water that each group of seeds gets. Now the next question, identify 2 conditions shown in the diagram that are held constant in all 4 pots. Again, I would call these constants, but you could also call them controlled variables. Number 2. Well, there's lots of possible answers here. The amount of soil each gets 4 seeds. They each get the same kinds of seeds. They're each put in the same sunny location. They've got the same kind of containers, same temperature. You get the idea. Everything else is kept the same except for the manipulated variable, which is the amount of water. That makes it a good controlled experiment. Okay, this one is from the 2013 Living Environments Regents exam, and I think this is a good one to really talk about and bring home some of the ideas. If you're looking at this on a small device, you probably can't read this, so again, I'll read it to you. Base your answers to questions 61 to 64 on the information below and on your knowledge of biology. Scientists have been experimenting with different forms of alternate energy to help reduce the amount of fossil fuels that are burned. They studied yeast, which convert plant materials into ethanol, a form of alcohol that can be used in automobiles. These experiments were carried out at room temperature. The scientists wondered whether more ethanol would be produced at different temperatures. It then asks you to design an experiment to determine the effects of temperature on ethanol production by yeast. In your answer, be sure to, and we've got 4 bulleted items here that should go in our answer. Now, you can either answer this in essay form and make sure that you answer each part in your essay, or it's perfectly okay to answer each bulleted item separately and treat it like a separate question. Um, that a lot of students like to do it that way because they're then sure that they've answered all of the parts. And that's the way I'm going to answer it this time. So the first bullet says state one hypothesis the experiment would test. What would you say would be a good hypothesis? Here's the one I came up with. Now there's more than one correct answer to this. Um, But if you read mine, you'll see I said if I increase the temperature, then yeast will produce more ethanol. Notice that I put it in the form of an if-then statement. Now the next bullet asks, state how the control group would be treated differently from the experimental group. If you remember, the control group is my normal setup, the one that is not being tested. And very often the control group would not, you would leave the independent variable out. Now in this experiment, my independent variable is the temperature, and you can't leave temperature out. That's impossible. You can't have no temperature. So to be normal, maybe I would say the control group would get um, room temperature. And then I would have some temperatures above room temperature and below room temperature to see if my hypothesis is correct. The next bullet asks to identify two factors that must be kept the same in both the experimental and control groups. These would be our constants. Well, there's a lot of good answers to this, but basically everything should be the same except for the temperature. Two constants that should be kept the same in all of the experiments and the control would be the amount and type of yeast that was put in each setup and the amount and type of plant material that was given to the yeast to be converted into ethanol. And finally, we need to identify the independent variable in the experiment, which we've already said. The independent variable, the one thing that we are changing or manipulating in the experiment, is the temperature the yeast are growing in. Okay, that's it for today. We'll see you all in class tomorrow.